energized or active. Okay, so what happens is now the second component of the nucleotide, as I said, was the sugar bases. Now here the purine nucleotides, they react with a sugar to form the respective nucleosides. Okay, and we know that the sugar in RNA is D-ribose, and that in DNA is again 2-deoxy D-ribose. Here I have circled the oxygen present in RNA, and I mean the RNA and DNA, you lack the particular oxygen on the second carbon. Okay, now how are these nitrogen bases and linked to sugar? It is always through the N-glycosidic linkage. There, when we had studied disaccharides, there also it was a glycosidic linkage only, but it was not N, it was O-glycosidic linkage, because two monosaccharides, they were linked together by a single oxygen atom. So there it was O-glycosidic linkage. Here it is N-glycosidic linkage, because the nitrogen is involved here in forming the bond with the sugars. So what happens is, here the ninth nitrogen of the purine ring is linked with the C1, first carbon of the sugar. Okay. So depending upon the nitrogen base present, when we only when we want to just name the excuse me, the nitrogen base, we call it as adenine, guanine, cytosine, thymine. But when you have a nitrogen linked to a sugar, then it becomes nucleoside, which are named differently. We call adenosine, guanosine, inosine, and xanthosine. Okay, and corresponding, if it is a deoxy, it becomes deoxyadenosine, deoxyguanosine, which is present in DNA. Now, this nucleoside is linked to the third component of the nucleotide, which is the phosphate group. Okay, the hydroxyl group on the fifth carbon. There, when the N-glycosidic linkage was formed, the first carbon of the sugar was involved in bond formation. But here, in what you call formation of the nucleotide, ultimately the OH group on the fifth carbon, okay, is linked with the phosphate group. I'll come to the structure. It will be very clear from here. Okay, if you can see,
I think he has some network issue. Okay, wait 2 to 3 minutes, then we'll start.
Shavni, where did you all get disconnected? At what point?
No, sir, you have range issue, sir, I think.
No. Where did it get disconnected? I'm asking which point. Hold on,
sir,
I'll share the content.
Nucleotide structure, sir.
This one?
Yes, sir.
Okay, so nucleoside, you all have understood. So that you all could catch up with.
Yes, sir. You have to explain phosphodiester bond, sir, right now.
Okay. So this page you all have seen, right?
No, sir.
Okay, wait. Then I'll talk. What about this one?
Yes, sir. It covered, sir.
Okay. So what as I said, in the formation of the glycosidic bond, here the nitrogen, the ninth nitrogen of the purine ring is linked with the C1, the first carbon of the sugar group. Okay, so that is why it is called as N-glycosidic linkage. On the contrary, as I said, in the disaccharide formation, it was a O-glycosidic linkage where one oxygen was involved in binding or involved in bringing two monosaccharides together. Now, coming to the phosphoester bond. See, now this is the structure of ATP, where this is the adenine. Okay, the adenine plus sugar you call it as adenosine. Okay, so if you all remember the numbering, this was the ninth carbon of the purine ring, which is linked with first carbon of the sugar moiety. Okay, and the fifth carbon here was free. But now what is happening in the during the process of formation of the NTP, here there are three phosphoric moieties which are linked with the fifth carbon. If you see the cursor moving, that is actually fifth carbon of the ribose sugar. Okay, and the bond which is formed between the sugar and phosphate, it is called phosphoester bond. Okay, phosphoester bond. And the bond between two phosphoric phosphate groups, it is phosphoanhydride bond. Okay, don't confuse phosphoester bond with phosphodiester bond. I'll come to that later. Phosphodiester bond is formed when two NTPs are linked together. Okay, here this is structure of a single NTP. NTP in the sense nucleotide triphosphate. Okay, so here what happens is the OH group on the fifth carbon of the sugar reacts with phosphate, and hence they can be called as 5' nucleotides. The bond between one sugar and phosphate is phosphodiester bond up to 3. Okay, this is all very to the what you call the overall charge. Now suppose if you see structure of ATP, when it is existing in cells under the physiological pH, the overall charge on ATP will be 4, because here there is one oxygen which is free, one oxygen is free, one oxygen free here, and one oxygen free. Because the valency of oxygen is 2, it can form 2 bonds. But here, since all are free, they will give an overall -4 charge to the NTP. Okay, but in the course of polymerization, the one of the phosphate groups will again be linked in formation of a bond. So therefore, it becomes -3 at the end. Okay, can you hear me?
Yes, sir.
I'm sorry, I'm unable to
Yes, sir. Yes, sir. You are
track
audible, sir.
the issue, whether it is from my end or it is from the other
No,
end.
sir. No, sir. Network issue from our side also, sir. No issue, please.
Because chalo, theek hai. So this did you all follow?
Hi, sir. Continue, sir.
Okay, fine. theek hai. So now
From
coming
ATP
to,
charges.
okay, so as I said, the overall charge on ATP or any NTP at physiological pH will be 4. Now, if you want to know the labeling of the phosphates also, so for the ease of understanding, they have been labeled as the first phosphate linked to the sugar. It is considered to be as alpha, beta, and then gamma. During the process of polymerization of these NTPs, the beta and gamma phosphate is released. Okay, which is what I said, ATP. On hydrolysis also it becomes ADP and PPI. And here in this process, 2 phosphates are released. So one is retained for forming the phosphodiester bond. Okay, so these are the various purine nucleosides which you all can easily understand. Now coming to the pyrimidine nitrogen base. Here also they are heterocyclic, aromatic, and planar. But the only thing is they lack the imidazole ring. Okay, and any doubt? Okay. So they lack the imidazole ring, and here the labeling, the numbering of the atoms will be in clockwise direction. Okay, so here it will be 1, 2, 3, 4, 5, and 6. I didn't label it in the structure, but the cursor where you can see the nitrogen cursor moving, that is the first nitrogen, second carbon, third nitrogen, fourth carbon, fifth carbon, sixth carbon. So that ways you have there are 3 types of pyrimidines, just like purines. You have uracil, cytosine, and thymine. Uracil, it only occurs in the RNA, whereas cytosine, thymine, they are found in DNA. Okay, uracil is again 2,4-dioxy pyrimidine. Cytosine is 2-oxy-4-amino pyrimidine, and thymine is 5-methyl-uracil, you can call it. Otherwise, it can be called 2,4-dioxy 5-methyl pyrimidine. Okay, so in this, in pyrimidines also, the nitrogen base reacts with sugar to form the respective nucleosides, and phosphate esters enter to form the nucleotides. But here what is happening is the glycosidic bond between the nitrogen base and sugar is that there the 9th carbon was involved in linkage. In case of purines, here the first nitrogen will be involved. This is the first nitrogen involved in linking with the first carbon of the ribose ring, and the NTP's story will remain the same. Okay, so coming to uracil, I mean the nucleoside and nucleotide forms, y'all can just go through it. Is not very difficult to understand. Now coming to formation of these phosphodiester linkages, you find them in oligonucleotides or polynucleotides of both DNA and RNA. Here what happens is it is formed, as I said, when the alpha phosphate of the successive nucleotide reacts with 3' hydroxyl group of the preceding nucleotide. Now this 3' hydroxyl group comes from where? It comes from this sugar. Okay, now see, you just read the statement here. It is formed when alpha phosphate, this was the alpha phosphate of the successive nucleotide, okay, which is linking with the 3' hydroxyl group. This was first carbon, second carbon, third carbon. So this is the third hydroxyl group, and they are involved in formation of the phosphodiester bond. That means 2 sugar units linked by a common phosphate group. This is phosphodiester bond. There, when you link the single sugar unit and the phosphate, that is the bond between oxygen and oxygen of the sugar and phosphate, that is single, that is phosphoester bond. Okay, you know what are esters, right? It is formed when acid and alcohol come together. Okay, so similarly, इसमें CH2OH group होता है sugar में, and here phosphoric acid. So acid and alcohol, when they come together, they form the ester, which is why these are named. So phosphoester bond and phosphodiester bond. These are the various ways of representing. And we know that the direction and sequence of nucleosides in any polynucleotide strand of RNA is always from 5' to 3' direction. We always name it as 5' to 3'. Okay. Please, you can just go through. Coming to the conformations. Okay. Now, just like sugars also, they were existing in different types of configurations. Here, these are the conformations. Okay. So because here what is happening, as I told you all before also, configuration and conformation. Conformation will not require any what you call it require it, I mean it requires what you call cleavage of the bonds, whereas conformations they do not require. So here also the nucleotides and nucleosides exist in two forms. One is the syn form and the anti form. Okay, I'll just show. Okay. So here in syn form, what happens is you'll have to imagine that the pyrimidine ring and the furanose ring. Furanose ring, that is the ribose ring of the sugar, they are present towards the same side, leading to steric hindrance. Okay. In anti, it is the opposite. Okay. Now I'll come to the figure. If you can see here the pyrimidine ring, okay, and the furanose ring. If you can see, they're on the same side. They are on the same side. So this can result in steric hindrances. Okay, but and in the anti-conformer, if you see the pyrimidine ring here and the furanose ring, they are slightly far away, or you can say opposite side of each other. So this prevents any kind of a steric hindrance. But for converting the syn to anti, it is just not rotation of the bond, it requires bond cleavage. Okay, which is why these are called as the configurations, syn and anti configurations, because just by rotating the ring, it will not it does not convert from syn to anti or anti to syn. Okay, so for example, if you see, this is a purine. Okay, so for purine, what happens is the syn mostly the syn conformer of the purine nucleosides are for most of the nucleotides and nucleosides here, the anti-conformers are more stable, as I said, because it prevents any kind of steric hindrance. Okay, but except for thymine. Thymine is more stable in the syn conformer. Okay. Now this is again the syn and anti-conformer of a pyrimidine molecule nucleotide. For example, this is the uracil, that is 2,4-dioxypyrimidine, which is linked to the sugar unit. And since here it is the orientation or structure is with respect to the second carbon. If you can see nitrogen, this is the first nitrogen and second carbon. Here they are on the same side. The furanose ring and the oxygen is present on the same side, which is why it is the syn conformer. Whereas here they are on the opposite side, which is called as the anti-conformer. Okay, but with respect to deoxy, as I said, most of the nucleotides, they are stable in the syn, but except for except for thymine. Why it is stable in the syn conformers is because of the methyl group. Okay, the methyl group in syn conformer in anti conformer can cause steric hindrance here. Okay, with respect to deoxythymine, the syn conformer is more stable as the methyl substituent on C5 is present away from the furanose ring when compared to the anti-conformation, and that will avoid any kind of a steric hindrance, which is why the syn is more stable in case of thymine only. Okay, now coming to tautomerization, I had explained about this even during the endiol formation of sugar residues when they are placed in acidic alkaline conditions. So due to tautomerization, there exists two different forms of these sugar, I mean nucleotides. One is the keto form and one is the enol form. Keto form, it is very well evident because it is present in the keto, keto group. This is the ketone group. Here, when you link the hydrogen, it becomes the enol form. Okay, these are also called as the lactam and the lactam forms of nucleotides. Okay, and coming to the any doubt in this?
No, sir.
If you all have, you all can just get back to me. And coming to physicochemical properties, then we know that even like just like sugars, nucleotides also contain chiral carbons. So they have this optical activity because owing to the sugar also which is present. Okay, then they absorb UV at 260 nanometers, and these nucleosides and nucleic acids, they are they are acids, and the presence of the phosphate group as a component makes them acidic. Okay, and they have the tendency to lose protons at physiological pH, which is why I had told that ATP at physiological pH will have overall negative charge, because they lose the protons very easily. Okay, so nucleotides and nucleic acids, they are negatively charged, and uh, hence DNA, which is gets associated with most of the histone proteins, which contain positively charged amino acids, which is lysine and arginine. So this is the basic what you call structure and functions of the nucleotides and nitrogen bases, their synthesis, where they are derived from, what happens during the metabolism, and the inborn errors I'll take up in when I start metabolism part of the biomolecules. So if you all have any doubt in this, you all can raise your doubts in the chat box, or you can ask me here. For before you all start, for examination point of view, that's what I told, the methylated xanthine derivatives. Okay, that is important, and the names of the nitrogen bases. Okay, what is adenine, what is guanine, what is cytosine, what is thymine. Okay, and one thing is which nitrogen and which carbon is derived from there. So that I'll cover. So these points you'll have to remember. And syn anti-conformations, if you can remember, it is well and good. But only which one is more stable, that you all should know. And the bond formation, that is important. Which carbon is linked with which nitrogen in purines, and which carbon is linked with which nitrogen in the pyrimidines during formation of the nucleoside. Okay, if you'll have anything, yes, I'm open for discussion. You all can put your message in the chat box also. So if you all don't have any doubts, then sir, what is the plan, sir, now? Shravani, can you please coordinate?
Hello, sir.
Yeah.
Sir,
So
if they
you all
don't have any doubt, we can conclude, sir.
We can conclude. Okay, in case you all go through the content, and you all can raise your doubts. Okay.
Okay, sir.
This was very small,
Sir,
actually, for exam. Bolo.
Sir, next topic is what, sir? Metabolism?
Next, actually, no. I'll finish proteins and lipids also. Okay, sir. But I'll require one day for that. I mean, 2 hours proteins and 2 hours lipids. Then metabolism also will take a long time, but that I'll just brush up quickly. Okay, sir. Because
so next mostly, next it may be proteins or lipid topics, sir.
So Tuesday, I think sir has not tomorrow. No, tomorrow I think sir has arranged one. But tomorrow, because tomorrow morning I have classes in IRI also. So morning I will not be free. Second half I have asked sir Plus, I've got one national workshop also to attend online. Anyways,
Okay, sir.
but I can take time. But that's what my request is. Please, y'all, if y'all can join by 3:30, it would be convenient for me also. Okay. I can
Okay, sir.
understand students may be attending other classes, but 5-10 minutes is
Even
okay.
now mid exams also going on, sir.
Yeah, I know. Students, I can understand. So
Yes.
no issue. If this I'll share the content at the end. Once I'll make a complete PDF and I'll share it with Dr. Bhalera, because I don't want to keep sending a single PDF every time.
Okay, sir. Thank you, sir.
Okay. Okay, fine. Thank you, Sharmi. I'll
Okay.
stop then.
Okay, sir. Okay, sir.